Hi, good evening. Can you hear me? Am I audible?
Yes, sir, audible, sir.
Yes, thank you.
Actually, I unmute the other students, sir. They will message you in chat.
Chat.
Sir.
Okay. Okay, sure, sure. Okay. So, good evening. So, I will start my session in a short while. So, since the chat box is active, so you can post your questions in the chat box. Okay. So I will start with my first question. So, myself, I am Dr. Jagdish. I am an associate professor working in Velamal. I did my MD in CMC Vellore. So, hope this session is going to be useful for you. So let's see. So if you have any doubts, you can just put it in the chat box. We can discuss it at last. Is that okay? Okay, just click say okay or no in the chat box. Okay, fine. In between, if I'm not audible or if what I'm speaking is not able to be understood, please say it. Okay. So you can just say that you can't understand. So we can revisit the slide or we can revise the concept again and then we can go to the next slide. Okay, fine. Okay. So let me start. So the first question, 1 minute. So the first question is this. So the first question is a 2-year-old boy was brought by his mother to pediatric OPD with complaints of difficulty in identifying the objects. The boy was found to have cataract in his right eye and his liver function tests were deranged. Identify the enzyme most likely to be affected. So, from the clinical case that has been mentioned here, two things are obvious. One, the boy is having some problem in his vision, and his liver is not normal, as suggested by the deranged liver function test. And what is the cause for his vision problem is cataract. So, he's having cataract and a liver problem. So, what do you think the answer could be? Okay, fine. Okay, so let's see. See, this is a classical picture of a defect in galactose metabolism. But the catch is here. See, this is how galactose gets metabolized. There are three enzymes responsible for galactose metabolism. One is called galactokinase, which converts galactose to galactose 1 phosphate. This is the first step. The second step, the galactose 1 phosphate will be converted to UDP galactose. So UDP is getting transferred. So it's called uridyl transferase. So galactose 1 phosphate uridyl transferase, and this UDP galactose is converted to UDP glucose. That is done by epimerase. These three enzymes, when it goes for a defect or when there is a mutation affects any of these enzymes, there could be a defect in the metabolism of galactose. All these three will cause cataract. So why do you say it's A? That's because galactokinase deficiency is called galactokinase deficiency. If galactose 1 phosphate uridyl transferase deficiency, it's called as classical galactosemia. See, what happens when galactose accumulates in the cell? So excess galactose is converted to galactitol. Okay. So I'm telling you why there is cataract if there is defect in any of the enzymes involved in galactose metabolism. So galactose converts itself to galactitol. And this galactitol is osmotically active. So it draws in water into the lens cell. So the lens will swell and the cell will damage, leading to cataract. So cataract is a defect in the lens, so that the transparency of the lens is lost. The lens will become opaque. So that's the opacity is due to the accumulation of galactitol, which is osmotically active and draws in more water. So difference is here. If galactokinase is deficient, the first step is not going to happen. So galactose accumulates. Okay. So galactose accumulates, as we saw in the earlier slide. Galactose accumulates, leads to galactitol formation, leads to cataract. This is pretty straightforward. So what happens in classical galactosemia, where there is a defect in the UDP galactose 1 phosphate uridyl transferase? So here, galactose 1 phosphate accumulates as well as galactose accumulates. So this is the difference. Okay. So increase in galactose leads to cataract, straightforward. And accumulation of galactose 1 phosphate can lead to inhibition of gluconeogenesis. Okay, first point. Second, since this step is defective, ATP will be consumed for converting galactose to galactose 1 phosphate. So it leads to depletion of ATP. So these two conditions will lead to liver damage. Okay, so that's why galactose 1 phosphate uridyl transferase leads to cataract as well as liver damage. So that's why this boy has cataract as well as his liver enzymes are deranged. So the correct option is option A. Okay, fine. So I'll just ask you here. So that's the first MCQ we discussed. Am I very fast, or is this speed okay? Are you able to understand something, or do you want me to decrease my speed? Is the speed okay? Okay, fine. Okay. So I have the habit of speaking fast, so that's why I'm asking. Okay, since we'll go to the next MCQ. So galactose metabolism in general will lead to cataract, but the presence of liver damage points a diagnosis towards classical galactosemia, which is caused due to the defect in this enzyme called galactose 1 phosphate uridyl transferase, which is also called as GalPut. So they can ask you what is GalPut. They should not ask, but if they ask you what is GalPut, GalPut is this: galactose 1 phosphate uridyl transferase. Let's go to the next MCQ. Okay. Glycogen stored in the skeletal muscle cannot contribute to blood glucose. Why? So there are four enzymes listed as options: glycogen phosphorylase, debranching enzyme, glucose 6 phosphatase, acid maltase. Before going to the correct answer, what are all these four enzymes? They are part of which pathway? Can you please type in the chat box? These enzymes are part of which pathway? No, it's not part of glycolysis. Okay, good, good. So there are 34. Only four are playing. Hmm. Okay, fine. So all these enzymes are part of a pathway which is involved in breaking down glycogen. Okay, so all these four enzymes are part of glycogenolysis. In this, one enzyme is also part of gluconeogenesis. What is that enzyme? Good. So glucose 6 phosphatase is an enzyme which is involved in gluconeogenesis. Good. Fine. Let's see why skeletal. See, glycogen is stored in skeletal muscle. It's also stored in the liver. But why only the liver glycogen contributes to blood glucose? Why not the skeletal muscle glycogen is not contributing to the glucose? The answer is here. So, see, in liver, glycogen breaks down to form glucose 6 phosphate. And this glucose 6 phosphate can be converted to glucose in the liver itself by an enzyme called glucose 6 phosphatase. However, in skeletal muscle, glycogen breaks down to glucose 6 phosphate, but skeletal muscle lacks this enzyme called glucose 6 phosphatase. So, skeletal muscle cannot convert glucose 6 phosphate to glucose. So, what happens to this glucose 6 phosphate, which cannot be contributed to glucose? So, why does it breaks down in the muscle? So, what's the purpose of glycogen breaking down in the muscle? The answer is simple. Okay. So muscle also needs energy, right? So the glycogen stored in the muscle breaks down to form glucose 6 phosphate, and this glucose 6 phosphate is converted to enters into glycolysis, which produces ATP. So that's the reason. So glycogen stored in the liver can contribute to blood glucose, but not the muscle, because muscle doesn't have this enzyme, glucose 6 phosphatase, which is responsible for converting the glucose 6 phosphate to glucose. So glucose 6 phosphate directly enters into glycolysis for ATP, so that muscle can use up the ATP and get its energy back. Okay, so glycogenolysis happens in the skeletal muscle mainly for its own energy purpose. It will not contribute to blood glucose because it doesn't have that enzyme called glucose 6 phosphatase. So that's the answer. So the correct answer is C. So third, very interesting question. Which of the following is true regarding gluconeogenesis? So what is the answer? Which of the following is true regarding gluconeogenesis? So one question is what happens in von Geerk's disease? I will come to that later. So what is the answer for this? Which of the following is true regarding gluconeogenesis? It's slightly confusing question, but you have to think a little bit. I'm asking which is a correct statement. Okay, most of them are saying A. Let's see whether A is correct or not. Okay, fine. See, gluconeogenesis. What is gluconeogenesis? Gluconeogenesis, if you see the definition, it is actually synthesis of glucose from non-carbohydrate sources such as lactate, glycerol, propionate, etc., etc. That is, whenever your body needs glucose, glucose can come from only two sources. One, whatever is stored as glycogen has to break down to form glucose. That's the first source. Second, the body should produce glucose. That is called gluconeogenesis. So, what are the substrates for gluconeogenesis? These are the substrates: propionate, lactate, glycerol, pyruvate. These are the sources of substrates for gluconeogenesis. So, can someone tell me what pathway is this? This the pathway which you see in your screen. What pathway is this? Yes, correct. It's glycolysis. Okay. See, if you see carefully, there are three reversible steps. Okay. The first step is exokinase step, that is irreversible. Phosphofructose kinase 1, which is a rate limiting enzyme of glycolysis, that is also irreversible. And the last step, pyruvate kinase, that is also irreversible. So there are three irreversible steps in glycolysis. So you cannot simply reverse this pathway to produce glucose. So gluconeogenesis is not an exact, simple reversal of glycolysis. You understand now? So there are three irreversible steps in glycolysis. One is exokinase, phosphofructose kinase 1, and pyruvate kinase. All these three enzymes are irreversible. In that, PFK1 is the rate limiting enzyme. So you cannot simply reverse it. So you have to bypass these steps. Only then you can go back to glucose. So that is what is gluconeogenesis, right? You have to produce glucose. So you have to bypass these steps. So how it is done? Pyruvate is converted to phosphoenol pyruvate first. Okay. So that is done by two enzymes. One is called pyruvate carboxylase. The other one is called PepsiK or phosphoenol pyruvate carboxykinase. These are the first two enzymes of gluconeogenesis. This step requires ATP. We'll see this in detail in the next slide. Okay. So this step requires ATP. And this step, PFK1 is reversed by fructose 1,6 bisphosphatase 1. So what could be the third enzyme, which we saw earlier? Glucose 6-phosphatase. Okay, these are the four enzymes of gluconeogenesis. Okay, so by this we can tell gluconeogenesis is not the simple reversal of glycolysis and it requires ATP. So we need ATP to reverse or to produce glucose from non-carbohydrate sources. So gluconeogenesis requires ATP. It is inhibited by insulin. How? Because insulin is a hypoglycemic hormone. Okay. So insulin will do everything to bring down the glucose levels in the body. So this pathway is able to produce glucose. So insulin has to inhibit this pathway. So insulin is inhibiting this gluconeogenesis by acting on this enzyme called phosphoenol pyruvate carboxykinase. Okay. First option. Okay, so as I told you earlier, we'll just see it in detail. So pyruvate is converted to oxaloacetate by this enzyme called pyruvate carboxylase, which requires biotin. So biotin acts as a cofactor for many carboxylases. One carboxylase is pyruvate carboxylase. And this step requires ATP. I told you earlier. Oxaloacetate is converted to malate by an enzyme called malate dehydrogenase. And from there, it enters into cytoplasm. Again, it's converted to oxaloacetate. Oxaloacetate is converted to phosphoenol pyruvate. Okay, so pyruvate is converted to phosphoenol pyruvate. So we reversed that step, which is catalyzed by pyruvate kinase. And this requires ATP. So pyruvate to phosphoenol pyruvate requires ATP. So this is evidence. So there are two enzymes involved: pyruvate carboxylase and phosphoenol pyruvate carboxykinase. I want all of you to remember this fact. And this enzyme called pyruvate carboxylase. What is the allosteric activator of this enzyme? Anybody? What is the allosteric activator of this enzyme? Acetyl-CoA. Okay, acetyl-CoA is an allosteric activator of this enzyme. That is, whenever there is a lot of acetyl-CoA in our body, it will convert pyruvate to oxaloacetate. So this enzyme as an allosteric activator is called acetyl-CoA. Okay, so what are all the sources of acetyl-CoA? One, it can come from fatty acid oxidation. Okay, beta oxidation of fatty acids produces acetyl-CoA. Okay, so whenever beta oxidation of fatty acids is occurring, so there is a possibility that gluconeogenesis can be activated. Okay, but it's because of this fact. So acetyl-CoA activates pyruvate carboxylase. Okay, fine. So let's come to the first option. So this is glycolysis. As I told you earlier, this is glycolysis. So pyruvate is converted to oxaloacetate, oxide is converted to phosphate. This is done by an enzyme called PepsiK, and this can be converted back to glucose. So whatever green you are seeing, green arrows suggest gluconeogenesis. Whatever red arrows you see, that is glycolysis. Okay. See what happens if the cell has a lot of NADH. Two things will happen. Pyruvate can be converted to lactate. Okay, so a lot of NADH is there. Pyruvate can be converted to lactate. So can anybody tell me what is the, sorry, can you still see my slide? Can you still see my slide? Oh, okay, fine. So what happens when there is, so this is what happens. Okay, so when there is a lot of NADH, pyruvate will be converted to lactate. Okay, what is the name of this enzyme? It's just this step. Pyruvate is getting converted to lactate. Anybody? What's the name of this enzyme? This enzyme is part of anaerobic glycolysis. No. Correct. This enzyme, the enzyme which catalyzes this step is called lactate dehydrogenase. Very good. Okay. So first, whenever there is a lot of NADH in the cell, the pyruvate will be converted to lactate. Second, the oxaloacetate will be converted to malate. Okay, fine. So we are losing two substrates now for gluconeogenesis. So pyruvate is not there because all of them are converted to lactate. Oxaloacetate is not there because it's converted to malate. So the gluconeogenesis will be inhibited because we need oxaloacetate to produce phosphoenol pyruvate. We need pyruvate to produce oxaloacetate. So both these substrates of gluconeogenesis lost whenever there is increased NADH. So gluconeogenesis is inhibited whenever there is increased levels of NADH in the cells. Okay, can you name me one condition where there is increased levels of NADH in the cell? When will a cell have a lot of NADH? I will tell you. See, whenever there is increased intake of alcohol, okay, alcohol, whenever there's increased intake of alcohol, see, one molecule of alcohol when it gets metabolized to acetate to form two NADH. So your NADH levels in the cell will increase. Okay. So alcohol will lead to increased levels of NADH. So increased levels of NADH will inhibit gluconeogenesis. That's the reason why alcoholics, whenever there's a binge alcohol intake, okay, somebody goes to a party, drinks heavily, the next day morning they'll come to casualty with hypoglycemia. This is the reason, because alcohol intake increases. Binge, especially binging, the binge alcohol intake leads to increased NADH levels, and this increased NADH will cause inhibition of gluconeogenesis because it diverts pyruvate to lactate and oxaloacetate to malate. So this is one more additional fact. So that's the explanation for this MCQ. So the correct answer is A. increased NADH-NAD ratio inhibits gluconeogenesis. Okay, fine. So hope you addressed all the options. I'll go to MCQ number 4. Okay, fine. So all of the following are correct regarding isoenzymes except. So this is an interesting question. Okay. So A, isoenzymes will differ in the stability towards heat. Second, they have identical electrophoretic migration. Third, they catalyze the same reaction. Fourth, they differ in their glycosylation pattern. Anybody? What is the answer for this? This is a very simple question, but other options are interesting. Okay, good, good, good. Okay, let's see. Let's see what is that. Let's see. Let's discuss about isoenzymes first. Okay, okay, fine. See, isoenzymes. These are the. This is the definition for isoenzyme. Okay, they catalyze the same reaction. Okay, so they will catalyze the same reaction. Second, but they differ in physical properties. But they differ in physical properties. Why they differ in physical properties? Because they are genetically different. Why? So one comes from one gene, the other isoenzyme can come from other gene. So when this happens, there will be difference in the amino acid sequence of that protein. Since because of this genetic difference, their amino acid sequence, see, enzymes are also proteins, right? Proteins are made up of amino acids. So because of this genetic difference, the amino acid sequence will vary in between one isoenzyme and another isoenzyme. So this is the reason why they differ in physical properties. Okay. So because of this, what are the difference in the physical? What are the physical properties by which they differ? First, they can move differently when you subject them to electrophoresis. What is electrophoresis? Electrophoresis is nothing but movement of charged particles from cathode to anode. And that is the movement of charged particles under the influence of electrical field. Proteins are charged particles. So they will move from cathode to, that is minus end to plus end. Okay. So this movement depends upon their molecular weight, size, charge, etc. Since they are different in their amino acid sequence, their molecular size also is going to differ. So they will move differently. So that's the first thing. They differ in their electrophoretic mobility, first point. Second, they differ in their susceptibility to heat. That means some enzymes, some isoenzymes are heat stable, some isoenzymes are heat labile. So some enzymes will tolerate heat, some enzymes will break down. What happens when you heat a protein? Or what happens when you heat a protein? What the protein will undergo? Anybody? Yes, protein will undergo something called denaturation. So when the protein is denatured, their biological function is lost because their tertiary structure, quaternary structure, secondary structure is lost. Only the primary structure will be intact. So when you heat a protein, it will denature. So when you denature something, it will not be able to move in the electric field. So the electrophoretic mobility is affected when you denature a protein. So some isoenzymes are susceptible to heat, some enzymes are not. Okay, so they differ in their susceptibility towards heat. Third, they differ in glycosylation. That is, because of the amino acid change, some will be glycosylated, some will not be glycosylated. So these are the three differences. Okay, there are other differences also which we will see in the subsequent slides. Okay, other minor differences are also there, we will see in the subsequent slides. Okay, so this is what happens. So what you see in the picture is one isoenzyme. What is this isoenzyme called? Lactate dehydrogenase, which we saw in the earlier slide, which converts pyruvate to lactate in anaerobic glycolysis. See, lactate dehydrogenase has five isoenzymes. It is made up of two subunits. A subunit A, subunit B. A is called H, B is called M. So it can be 4H, 3H, 1M, or 2H, 2M, like that. Okay. So if you see the electrophoretic migration, see LDH1 goes first, followed by LDH2, followed by LDH3. Sorry. Followed by LDH4 and LDH5. So that's how they are named also. So one which goes first towards the anode is called 1. One which is farther is called 5. Okay, so there is one important MCQ in this LDH isoenzyme pattern. Okay, there's something called flipped pattern. You know what is meant by flipped pattern? Flipped pattern of LDH, where you will see. What is flipped pattern? Correct, you will see it in MI. So usually LDH2 is more than 1 in concentration wise, I'm telling. But in MI, 1 will increase. So there will be a flipped pattern. Okay, so that's the thing. Okay, so that's one important MCQ regarding LDH. Okay, fine. So I told you what is susceptibility to heat. There's one more enzyme for which isoenzymes plays a very important role in diagnostics, that is alkaline phosphatase. Okay, so in which conditions we measure alkaline phosphatase too often? What are the conditions where we measure alkaline phosphatase? Alkaline phosphatase is a marker for cholestasis. Okay, what is cholestasis? It is a marker for obstructive jaundice. Okay, it's a marker for obstructive jaundice. Somebody said metabolic disease of bone. Yes, it is correct, but it's not that common. Okay, alkaline phosphatase is a part of liver function test. Okay, it is mainly to diagnose something called obstruction in the bile flow. Okay, it's a marker for obstructive jaundice. Okay, so this alkaline phosphatase exists in many isoenzymes, mainly 5 isoenzymes. You see on the right side, left side, there are 5 enzymes: intestinal alkaline phosphatase, bone alkaline phosphatase, placental alkaline phosphatase, liver cell alkaline phosphatase, and alkaline phosphatase from the biliary canalicular cells. Okay, and if you see their migration, intestinal ALP is heavy, so it stays in the gamma region. One which moves faster is the biliary ALP. That's why it's going near alpha 1. In between are the bone, placental, and liver cell. Okay, fine. So if you see the susceptibility to heat, okay, placental ALP is very, very heat stable. That is, if you take a blood sample, you isolate the serum, and if you pre-treat it, and if you heat them before you put them into electrophoresis, plus all isoenzymes will get denatured. It's a placental ALP because placental ALP is heat stable. So only that will move. So you'll see only one band. So that band is because of placenta. Okay, so that's one way of differentiating the isoenzyme pattern before and after heating. So that's the reason why they differ in their differ with respect to their susceptibility to heat. Okay, fine. So there's two more things which are relatively not common, but it's good to know from the MCQ point of view. Okay, so the alkaline phosphatase isoenzymes can also be classified based on their incubation with an enzyme called neuroaminidase. See, neuroaminidase is an enzyme which is responsible for removing something called sialic acids. Okay, so all isoenzymes except intestine, they are sensitive to this enzyme. So this enzyme can go and act on all these enzymes and remove the sialic acid. So when you remove something from a protein, so their molecular weight will change, right? So their migration will also will change. But the intestinal ALP is resistant, so it will not change at all. So it will remain in the gamma region. There's one more way of picking up the intestinal ALP from the serum. And there's a third one, which is called affinity towards lectin. Bone ALP is more affinity towards this lectin protein. So it binds to it. So it goes and binds to one more protein. So it becomes more heavy. So it will not move at all. So that is the other way of picking up bone isoenzyme from the blood circulation. Okay. So all isoenzymes are predominantly picked up by one technique called electrophoresis. Okay, based upon their rate of migration in the electric field. Okay, so you can subject them to electric field as such. What we do for LDH, no change. You will see 5 different bands: 1, 2, 3, 4, and 5. So the answer is simple. There is no problem at all. But in alkaline phosphatase, if you see, so even though they migrate to different regions, see they are not easy to identify. So you need further techniques to identify what enzyme exactly is present in the blood circulation. So you can subject them to heat because placental ALP is completely stable, so it will not get denatured at all. Okay, incubation with neuraminidase, intestinal ALP is resistant, whereas other enzymes are sensitive, so they will lose their sialic acids and they will move to different places and affinity towards lectin. So this is one important point. Okay, when it comes to alkaline phosphatase, there are 3 special isoenzymes. They're called Regan ALP, Nagao ALP, and Kasahara ALP. See, very simple. All these isoenzymes are elevated in patients with malignancy. Okay, so they have different origins, but These three isoenzymes of ALP are seen only in patients with malignancy. Okay, very rare, but they have names for it, and this will be interesting for the examiners to keep it as a question. Okay, so Regan, Nagao, and Kasahara, they are all isoenzymes of alkaline phosphatase seen elevated in patients with malignancy. Okay, fine. Okay, so what will be the answer? The answer will be B. So if they have identical electrophoretic mobility, that's actually not true because they move to different places in electrophoresis. Okay, okay, fine. So okay, so inclusion cell disease is associated with which of the following organelles? Answer is, what is the answer? Mm. What is the answer? A D. Why A and D? Confusing is it? A and D. Okay, fine. Okay, slow stage. Okay, fine. So answer is actually D, lysosomes. Okay, I can understand why people have said that A, but the more appropriate answer would be D. Okay, see, whatever proteins are usually synthesized in ribosomes, right? It is synthesized in something called rough endoplasmic reticulum. That is ribosomes attached to endoplasmic reticulum. There's one more in endoplasmic reticulum, smooth endoplasmic reticulum. Proteins are synthesized only in the rough endoplasmic reticulum. From ribosomes, from the endoplasmic reticulum, they should go to respective organelles. Okay, that is all proteins required for the cell are synthesized in the ER, and from ER, it has to go to plasma membrane because some protein should go to plasma membrane, some protein should go to mitochondria, some protein should go to lysosomes, some protein should go to peroxisomes. So they should go to different places. This is called protein trafficking or called protein targeting. Okay. So this protein trafficking will happen by means of a specific signal sequence which are added to them in the Golgi body during their synthesis. Okay, fine. So that is why I think many of them have answered A. Okay, so they are added, the signal sequence is added to this protein either in the ER or in the Golgi body. Okay, so for example, the proteins which has to go to lysosomes should have a signal sequence which is nothing but one molecule called mannose 6 phosphate. So if you want to send a protein to lysosome from the rough endoplasmic reticulum to lysosome, you have to attach mannose 6 phosphate to this lysosome. So if this mannose 6 phosphate is not attached to lysosome, it cannot go to lysosome. So it will go out of the cell. So it will go out of the cell. So what are these lysosomes consist of? They consist of mainly hydrolytic enzymes. That is, the proteins which are mostly present in the lysosomes are responsible for hydrolyzing the waste products, right? So these hydrolytic enzymes, instead of going to lysosome, will go out of the cell. So they'll enter into blood circulation. So lysosomes will lack this enzyme. What happens because of this? So the lysosome's main action is to degrade mucopolysaccharides, mucolipids, etc., etc. So this will not happen. So lysosomes cannot degrade these mucolipids and mucopolysaccharides. So this will appear like inclusions when you see in the biopsy. That's why it's called as inclusion cell disease. So this has many features such as coarse facial features. Patients with inclusion cell disease will present with coarse facial features, mental retardation, skeletal deformities. So it is mostly the answer is D because the proteins for the subject which has to go to lysosomes is not going to the lysosome. So it is coming out of the cell. So it's mostly associated with lysosomes. I can understand why people have said Golgi, but I would say the more appropriate answer would be lysosome because this is actually a lysosomal cell disease. Okay, this is called inclusion cell disease. Okay, let's see about the other options. For example, Golgi bodies. There's one disease called Pompey's disease. What is this Pompey's disease? Have you come across this Pompey's disease anywhere else? Yes, it's a glycogen storage disorder. Okay, it's a type 2 glycogen storage disorder. Okay, mitochondria, all these mitochondrial disorders. Okay, Leber's hereditary optic neuropathy. What is so special about mitochondrial disorders? There's one thing unique about mitochondrial disorders. What is it? If you draw a pedigree, you will understand about mitochondrial disorder. What is one thing? Yeah, it's maternal inheritance. Okay, it comes from mother. It affects only the girls. Okay, it is has mitochondrial diseases will exhibit maternal inheritance because all mitochondria is usually derived from the ovum, not from the sperm. Okay, so peroxisome, there's one disorder called Zellweger syndrome where peroxisome is not produced at all. Okay, there's one more disorder associated with peroxisome. I think we have an MCQ on this also in the coming. I think the next MCQ, Refson's disease. Okay, so there's a one protein which has to be targeted to peroxisome. Because of a defect, this won't go to peroxisome, and that leads to a condition called Refson's disease. Okay, fine. We'll see that in the subsequent slide. Okay, fine. So the answer is lysosome. The answer is D. Okay, it could not be A because it's close. I said it's closely associated. It's closely associated with the lysosomes, not B, not A, not Golgi bodies. Okay, fine. So what is the answer for this? Good. Refson's disease is associated with the defect in oxidation of phytanic acid. Good. So it's very good. That's the answer is phytanic acid. Okay. What is this phytanic acid? See, Refson's disease is actually it's due to a defect in an enzyme called phytanyl CoA hydroxylase. This enzyme is part of peroxisome. Okay, this enzyme is present in peroxisome. Okay, this enzyme, as the name suggests, it acts on an acid called phytanic acid, which is nothing but a fatty acid, which is a branched chain fatty acid, particularly found in milk products and animal fats. Okay, this phytanic acid has to be acted upon by this enzyme for further oxidation. But if you don't have this enzyme, this phytanic acid cannot be broken down. It cannot be oxidized. So these branched chain fatty acids will accumulate in the circulation. Okay, and they are all neurotoxic. Okay, the patients will present with neurological manifestations like hearing loss, visual disturbances. They can cause retinitis pigmentosa, peripheral neuropathy, and cerebellar ataxia. Okay, so that's the reason why it is called. They have neurological manifestation because of the accumulation of this branch chain fatty acids in the circulation. Okay, the skin biopsy will show lipid laden vacuoles. Why lipid laden? Because of this, the cells will have all those unoxidized branch chain fatty acids. They'll go into the cell, but it will not be oxidized because peroxisomes lacks this enzyme. So they'll go into the peroxisomes, but peroxisomes cannot be able to hydrolyze it because they do not have this enzyme. So the peroxisomes will have lipids inside it, which is nothing but these fatty acids. So skin biopsy will show this variably sized lipid laden vacuoles. So the answer is phytanic acid. Okay, so resistance is also is caused due to the different oxidation of some fatty acid called phytanic acid, which is a branched chain fatty acid present in the milk products and animal fats. If there is defect in the enzyme called phytanyl CoA hydroxylase, which is an enzyme present in the peroxisome. Okay, next. This is an interesting question. I don't know how many of you would have, I mean, most of you have already known it, but this is an interesting question. Okay. I felt this was interesting when I was setting this question. What was the answer? Okay. Most of them are saying A. Okay. Good. The answer is A actually. Correct. So what is this unripe lychee fruit? Okay. This has happened for real. Okay. This is not something I wrote it up. Okay. This happened for real in 2014 to 2019 in Bihar, Muzaffarpur, etc. etc. Okay. So on I mean I said etc. because all those regions were all those regions were having this public health epidemic going on. Okay, so the answer is A, correct. What is this? Is see unripe lychee fruit, especially when malnourished children. Okay, malnourished children takes this unripe lychee fruit. Can you see a lychee fruit there? That's how it looks. So this unripe lychee fruit will contain some chemicals. One methylene cyclopropyl glycine. Okay, methylene cyclopropyl acetic acid. The third one is called hypoglycine A. Okay, these toxins will inhibit beta oxidation of fatty acid. You know why? So if you remember the previous slides we discussed, you can answer this question properly. Okay, so we'll see how. Okay, so these toxins MCPG, MCPA, and hypoglycine A inhibits entry of fatty acids into mitochondria. For a fatty acid to enter into mitochondria, which shuttle is important? For a fatty acid to enter into mitochondria, which shuttle we need? Think and answer. See, for a fatty acid to get oxidized, it has to enter into mitochondria. So, which shuttle it is? Think. Good. At last, finally, one good answer is coming. One right answer is coming. Correct. Carnitine shuttle. Okay. For some reason, these toxins will inhibit the entry of fatty acids into mitochondria. One. Okay. Second, it inhibits one more important enzyme which is responsible for beta oxidation of fatty acid, which is called acyl CoA dehydrogenase. Okay. So these two things are inhibited by these toxins. When you inhibit these two, what is the ultimate effect? Beta oxidation cannot happen. Okay. So beta oxidation cannot happen. Okay. What's the ultimate end product of beta oxidation of fatty acid? When a fatty acid enters into beta oxidation pathway, what is the final product? Good. The final product is acetyl CoA. Good. Fine. So since these children, when they have this, when they have taken this lychee fruit, these toxins will be released into their blood, and that will inhibit the beta oxidation of fatty. So you are actually getting rid. So these babies cannot derive ATP from one important nutrient called fatty acid. So fats cannot help them to generate ATP because beta oxidation is inhibited. Okay, so they are completely dependent on glucose. They are completely dependent on glucose for their energy. So glucose, one, they are already malnourished, right? So their glycogen storage will not be that great. So they can give glucose only for a short period of time. Am I right? So glucose reserves will be lost. As you said, beta oxidation's product is acetyl CoA. If you remember the previous slide which we discussed, acetyl CoA is an allosteroic activator of pyruvate carboxylase. Correct. So there is no acetyl CoA. Pyruvate carboxylase cannot be activated. If pyruvate carboxylase cannot be activated, gluconeogenesis cannot happen. So you cannot utilize glucose because there's already less resource, and you cannot even produce glucose because gluconeogenesis is inhibited. So double blow here. So glucose is also cannot be produced. Beta oxidation of fatty acids cannot happen. So what happens? Complete hypoglycemia will happen. So that leads to brain damage. That leads to hypoglycemic encephalopathy. So this is the reason why there is death when especially malnourished children take this fruit, unripe lychee fruit. Okay, fine. I'll ask you one question with related to this. Have you ever come across this in a different name? Inhibition of acyl CoA dehydrogenase leads to a condition which has similar features in a different name. Correct. Sudden infant death is also correct. Okay. Any other name? Inhibition of acyl CoA dehydrogenase leads to this condition. What is that? What is the name of that condition? It is called unripe aki fruit. Both lychee and aki come from the same family. Unripe aki fruit can inhibit acyl CoA dehydrogenase and lead to something. What disorder is that? Anybody? Chris Gale comes from this place. Chris Gale, cricketer. Yes, it's called Jamaican vomiting sickness. Okay, that also has the same features. Okay, but only thing is that is Aki, this is Lychee. That's it. Both are one and the same, but both will have the same features. Okay, that is called Jamaican vomiting sickness. Jamaican vomiting sickness. So that is caused due to inhibition of acyl CoA dehydrogenase by ackee fruit. Ackee fruit also contains hypoglycemia A. It's called ackee, A-C-K-E, ackee fruit. Okay, Jamaican vomiting sickness. Okay, so that's why that's so that's about it. Okay, so unripe lychee can lead to these toxins, and these toxins can inhibit the entry of fatty acids into the mitochondria. It can also inhibit one more enzyme called acyl-CoA dehydrogenase. When both these are inhibited, it leads to inhibition of beta oxidation of fatty acids, and this will lead to hypoglycemic encephalopathy because they cannot use glucose and they cannot even produce glucose because gluconeogenesis is also inhibited because of lack of acetyl CoA. Okay, let's go to the next slide. So the answer is sorry, the answer will be MCPG, which is option A. Okay, fine. Next MCQ is this. Okay, fine. So I think we all know this. What's the answer for this? No, it's not D. Come on, everybody, type the answer. I could see only two options. Good. Okay, good. Good. Okay, fine. So the answer is A. Proprotein convertase subtilism kexin type 9. So also called as PCSK9. That is a protein which regulates recycling of LDL receptors to the cell surface. Okay, that is this protein has something to do with the recycling of LDL receptors. So what exactly is the role? So we'll see that. Okay, so this is a normal pathway. Okay, so these are the LDL receptors. So these are the LDL receptors which are coated by something called um, which are endocytosed by vesicle coated with clathrin. Okay, so the LDL receptors will take up lipoprotein, most probably LDL, that is low density lipoprotein. So it takes up inside endocytosis. So cholesterol, this endosome will fuse with lysosome. Cholesterol will be degraded, but the receptors will be recycled back to the surface. Okay, this is called LDL receptor mediated endocytosis. Okay, so there are LDL receptor takes up the cholesterol, which is LDL cholesterol, which is carried by LDL, gets inside by endocytosis. Endosome fuses with lysosome, cholesterol is degraded, but receptors is recycled back. Okay, this normally happens. Okay, receptor should go back to the surface. Only then the subsequent LDL will be endocytosed. So this is called as receptor recycling. Okay, what does this PCSK9 do? This is how what it does. Okay, if you see on the left side, LDL comes binds to the LDL receptor, which is a blue color one, and this binds to another protein called PCSK9. So vesicle goes inside along with PCSK9. So when PCSK9 is bound to LDL receptor, everything will get degraded, including the receptor. So including the receptor, everything will get degraded. So PCSK9, it is bound to this complex and when it gets endocytosed, everything will get degraded, including the LDL receptor. LDL receptor will get degraded, cholesterol will get degraded, and lipoprotein will get degraded. So cholesterol will be released into the cell, but lipoprotein also will be degraded. So receptor cannot go back to the surface. So PCSK9 will mediate lysosomal degradation of LDL receptor along with LDL. Okay, so is this good or bad? It is bad, right? Because we need a lot of LDL to be on the surface. Why? Because only if you have receptors on the surface, you can take up all the LDL cholesterol from the blood into the cell and degrade it. Okay, so LDL cholesterol is bad because LDL cholesterol can deposit in the blood vessels, leading to something called atherosclerosis. Okay, so we need a lot of LDL receptors to be on the surface. So it can take up all LDL cholesterol into the cell and degrade it. Okay, but this PCSK9 takes up the LDL inside and degrades it. So there's no LDL receptor at all. So we need to prevent this. So they give something called PCSK9 inhibitors. Okay, to treat hypercholesterolemia, especially familial hypercholesterolemia. Okay, so which already has an LDL receptor defect. So that's why the answer is A. So which enzyme is involved in the regulation of LDL receptor recycling? The answer is PCSK9. Okay, what exactly its role? It goes and binds to the LDL receptor complex along with and degrades it. So LDL receptor is degraded. So if LDL receptor is degraded, LDL cholesterol cannot come from the blood into the cell. So LDL cholesterol will be very high in the blood, which is very bad because it can promote something called atherosclerosis. So the answer is PCSK9. Okay, so this PCSK9 inhibitors is being used in practice, especially in those people who have very high levels of LDL cholesterol in the blood, and it gives very good results. It gives at least, as the literature says, PCSK9 inhibitors can bring down the LDL cholesterol by 40 to 50 percentage. The one drawback is that it is costly. Okay, hopefully the cost will come down soon. Okay, so that is one drawback of this drug. Okay, especially who have cholesterol, LDL cholesterol more than 300, 400, they will try this drug so that it will come down fastly. Okay, that is called PCSK9 inhibitors. Why we need to inhibit this protein? So that a lot of LDL receptor will go to the surface. Okay, so that's the answer for this MCQ. PCSK9 inhibitors. PCSK9 is a protein which is involved in regulating the recycling of LDL receptors to the cell surface. Okay, fine. A true statement regarding uncouplers. What is the answer for this? 9, it increases ATP synthase activity. B, it causes proton leak. C, there's one protein called uncoupler protein 1 present in white adipose tissue. D, it inhibits ATP ADP transporter. Okay, the answer is what? Come on, type. The answer is B. It's not B, it's B. Okay, so the answer is B actually. So we'll see how. Okay, so there's a whole electron transport chain. Okay, I'll just describe. Okay, this is complex 1, complex 2, complex 3, complex 4, complex 5. And there are two mobile electron carriers which are called coenzyme Q, which is called as ubiquinone. There's one more electron carrier called cytochrome C. Okay, NADH enters electron transport chain in complex 1. FADH enters electron transport chain in complex 2. From there, it donates electrons. It moves from 1 to Q, from Q to 3, 3 to 4, and at last, during each electron movement, 4 protons are pumped into the intermembrane space by complex 1, by complex 3, and only 2 protons by complex 4. Complex 2 will not pump any protons. Okay, so the protons are pumped only by 3 complexes. What are those complexes? Complex 1, complex 3, complex 4. Okay, complex 1 4 protons, complex 3 4 protons, complex 4 2 protons. So when the electron moves from complex 1 to 3 to 4 or 2 to 3 to 4, protons will be pumped into the intermembrane space. Okay, and this will create something called proton motive force, which will activate complex Y called ATP synthase, which will convert ADP and PA to form ATP. Okay, this is how electron transport is coupled with ATP synthesis. How electron transport is coupled with phosphorylation. How oxidation of NADH and FADH is coupled with phosphorylation. This is called oxidative phosphorylation. Okay, these two processes are always coupled. Electron should move, proton should be pumped, and this proton should go and activate complex Y and produce ATP. So these two processes should be always coupled. Okay, so there's one more important molecule that you should remember. There's a transporter called ADP ATP transporter. Okay, so which puts ADP inside in exchange for ATP. So you need ADP to be inside. Only then you can produce ATP. Okay, if there is no ADP inside, ATP cannot be produced. Okay, so this is a whole electron transport chain. Okay, fine. So this enzyme, this drug inhibits this transporter, ADP ATP transporter. Okay, so what happens in a brown adipose tissue? There is an uncoupler protein called UCP1. Okay, so this UCP1, what will do? It will introduce an alternate channel. That is, it acts as a transporter. It goes and sits in the inner mitochondrial membrane and forms a channel. When this channel is formed, all these protons, which is in the space, will enter through this channel into the matrix. So what happens? The proton motive force, instead of acting on ATP synthase, it enters into this matrix for nothing. Okay, so it just enters into matrix. Since a lot of protons are coming through this transporter, the energy of this movement is transmitted as heat. Okay, so ATP synthase will not function. So ADP will not function. So there won't be any function of ATP. Okay, so only few protons will come. So that is not enough to produce ATP at all. Okay, so that's what happens. So this uncoupler protein 1 produces a channel which you see in blue color here, and all proteins will come into the matrix through this channel. So instead of getting into complex Y, everything will leak into the matrix through this channel. So ATP cannot be synthesized. So ATP synthesis activity is decreased by uncouplers. Okay, and this uncoupler protein are mostly found in adipose tissue. Which adipose tissue? There are three types of adipose. What are three types of adipose tissue? We already know: white, brown. What's the third type? We have three adipose tissues: white adipose tissue, brown adipose tissue. What is the third type? What color you will get when you mix white and brown? Anybody? What will be the color when you mix white and brown? It will be light brown, right? It has a name. It's called beech. It's called beech fat. Okay. So there are three fats: brown fat, white fat, beech fat. Beech fat also has uncouplers. Okay. So brown adipose is a mains is a rich source of uncoupler protein, UCP1. Okay. So this brown adipose tissue is found mostly in infants, mostly in infants, especially in the interscapular regions. Okay. So these UP1s allows protons to escape, leak into the matrix instead of ATP synthesis, thus releasing heat. Okay, so it's very, very important for babies to maintain their temperature control. So brown fat is very, very important, especially in babies, to maintain the temperature. Okay, fine. Let's go to the next slide. Okay, so what is the answer? The answer is B. Okay, fine. 10. What's the answer? Acute intermittent porphyria is caused due to the deficiency of acute intermittent porphyria is caused by the deficiency of. Fantastic. The answer is D, hydroxymethylbilin synthase. We'll see how. Okay. See, one fact you should remember. Porphyria is due to the defect in the heme synthesis pathway. One point. Second point, there are a lot of steps. If the defect is in the early steps, it will lead to acute porphyria. Okay. This fact is very important. Okay. So even if you don't know, if you know the first, even if you know this fact, you can answer the question somewhat. Okay. So if the defect is in the earlier steps, it leads to acute porphyria. Okay. So porphobilinogen is converted to hydroxymethylbilane or uroporphyrinogen 1 by this enzyme called HMB synthase. One fact is that this enzyme has three names. Okay. So HMB synthase or porphobilinogen deaminase. So one amino group will be removed here. Since uroporphyrinogen 1 is synthesized, it's called as uroporphyrinogen 1 synthase. Okay, so they can ask you by three different names. Okay, so HMB synthase defect can lead to something called acute intermittent porphyria. Okay, so what are the classical features of porphyria? I mean, what are the classical clinical features of porphyria? Photosensitivity, good. One more important, abdominal pain, very good. Next. Correct. It has neuropsychiatric manifestations. Okay. So neuropsychiatric manifestations, photophobia, and abdominal pain. You know why? What is the reason? This PBG, no, porphobilinogen. Okay. This porphobilinogen is actually can act as a neurotransmitter. It's actually a hypothesis. It's not proven yet. Okay. ALA, amino levulinic acid, and PBG can accumulate in the skin. Okay, and the skin, when they get accumulated in the skin, when the sunlight falls on the skin, it can produce oxidative species, which can damage the skin cells. That is the reason for photophobia. Okay, and this ALA and PBG can act as neurotransmitter. Okay, that can cause abdominal pain and neuropsychiatric symptoms. Okay, so these are the biochemical basis for those symptoms of porphyria: abdominal pain, neuropsychiatric symptoms, photophobia, and hemolytic anemia. Hemolytic anemia because there is no heme at all, so there won't be hemoglobin, so there won't be any. There won't be hemoglobin, so there will be anemia. Okay, so these are the three main features. Peripheral neuropathy also, so neurological manifestations. These are because of the accumulation of ALA and PBG. ALA and PBG are like they are like neurotransmitters, so they can cause photosensitivity and they can cause neuropsychiatric symptoms. Okay, fine. So let's see about the other options. Okay, so when there is uroporphyrinogen decarboxylase, it leads to something called porphyria cutanea terda. It's a cutaneous porphyria. It affects mostly the skin. There won't be neuropsychiatric symptoms. If there is protoporphyrinogen oxidase, it's called variegate. Proporphyrinogen oxidase is called, the name says, hereditary coproporphyria. Uroporphyrinogen 3 synthase. What we saw earlier is uroporphyrin 1 synthase. This is 3 synthase. If this is 3, it is called as erythropoietic porphyria. Okay, that is congenital erythropoietic porphyria. Ferrochelatase is the last enzyme. Okay, this is the last enzyme in the heme synthesis pathway. It's responsible for introducing iron into the heme. Okay, this is the last enzyme. So when this last enzyme is defective, it is called as erythropoietic porphyria. Okay, fine. I know this porphyria is a little bit more of memory based thing because you have to remember all these enzymes. Okay. So but I think if you remember some facts, it will be easy. Okay, so let's go to the option. The option will be HMB synthase. Okay, so A will cause coproporphyria, B will cause porphyria cutanea tarda, C will cause variegate porphyria. Protoporphyrinogen oxidase will cause variegate porphyria. Okay, okay, 11. A 2-year-old child was admitted in hospital with jaundice, anemia, and loss of weight. Total bilirubin is 5. Indirect is 4.5. The enzymes are normal, and urine is positive for urobilinogen. So, what type of jaundice is this? Why post-hepatic? Hmm. So, which bilirubin is elevated in this? Which bilirubin is elevated? Just tell me which bilirubin is elevated. Unconjugated bilirubin is very high in this patient. Okay. So, out of 5 milligrams which you find in the blood, 4 milligrams are unconjugated. Okay. So, it's unconjugated hyperbilirubinemia. Okay. What about the enzymes? All enzymes are normal, right? So, there's no derangement in the enzymes. The enzymes are all normal and the urine is positive for urobilinogen. This is a classical picture of prehepatic jaundice. Okay. Why? Let's see. See, whenever prehepatic jaundice is also called as hemolytic jaundice, whenever there is hemolysis, okay, hemolysis is RBC breaks down, heme is released, heme will get converted to bilirubin. So a lot of heme comes out, so a lot of bilirubin comes out. Okay, so that bilirubin is called unconjugated bilirubin. Okay, and this unconjugated bilirubin should go to liver, where it gets conjugated and secreted into the bile, and that goes to the intestine. This is a normal pathway. Okay, since lot of unconjugated bilirubin is coming to the liver cannot conjugate everything, right? So the conjugation capacity of the liver will get saturated. So it can no longer conjugate. So it will reach its maximum. That's why a lot of conjugated bilirubin will enter into the liver. Okay, one. So beyond a certain point, it cannot conjugate at all. So unconjugated will remain as unconjugated. So indirect bilirubin will be very high in the blood. Okay, since lot of conjugated comes into the intestine, this conjugated bilirubin is the one which gets converted to urobilinogen. Okay, so urobilinogen will be positive or it will be increased in patients with hemolytic jaundice. Okay, so this is a classical picture of hemolytic jaundice or prehepatic jaundice. Why? Because serum bilirubin total is elevated. First point. Second, indirect is very, very high. because out of 5 milligrams that you see in the blood, 4 milligrams are unconjugated, which is nothing but indirect. Okay, that's the first point. Second, indirect is one which is slightly higher. Urine bilirubin is absent, which I didn't give you in the case history, but I have given you this. Urine urobilinogen is very high. Correct. That's because of this fact. Okay. And ALT, AST will be normal. ALP will also be normal. So the clinical case which has been mentioned in this previous slide is a classical picture of prehepatic jaundice, also known as hemolytic jaundice. Okay. Okay. Since there was a confusion, I'll show you what is post-hepatic also. Okay. See, post-hepatic jaundice is because of the biliary obstruction. I told you earlier itself. Okay. So because of the biliary obstruction, what do you see here? Can you see my cursor there? Can you see my cursor? There's a pointer there. Okay. So there is a block in the biliary tract. Okay. So conjugated bilirubin cannot enter into the intestine. So from the liver, it cannot enter into the intestine. So that's a normal flow, right? So since because of the block, the conjugated bilirubin or the bile will stay in the liver itself. So the conjugated bilirubin will leak into the blood circulation. Okay, so then the conjugated bilirubin will be very high in this patient, in these patients. So this is a classical picture of obstructive jaundice. Okay, because of the obstruction, the liver which conjugates the bilirubin and that bilirubin cannot enter into the intestine. So this bilirubin will leak into the blood circulation. Okay, so the conjugated bilirubin will be very high in patients with obstructive jaundice. Okay, fine. So in patients with obstructive jaundice, total bilirubin will be high. Indirect will be normal. I'll address that. So indirect will be normal or slightly elevated. Okay, direct will be very high. Urine bilirubin will be present because conjugated is water soluble. It comes out in urine. So urine bilirubin will be present. Urine urobilinogen will be absent because if the bile cannot enter the intestine, so there is no source for urobilinogen at all. You cannot produce urobilinogen at all. So urobilinogen will be absent or decreased, and AST-ALT will be slightly elevated, but ALP will be very high because ALP is a marker for biliary obstruction. Okay, you understand? This is a picture of post-hepatic jaundice or obstructive jaundice. Okay, so I think I answered the Harinandan's question. So what happens to urobilinogen in post-hepatic? It won't be there because urobilinogen has to be produced in intestine. Okay, it has to be produced from the intestine to enter into the intrahepatic circulation, and some more of it will enter into the kidney. Okay, to go to the kidney through blood circulation. So the source is conjugated bilirubin. So if you don't have conjugated bilirubin, urobilinogen will not be produced. So urobilinogen will be absent or decreased in patients with obstructive jaundice. You understand? Okay, fine. So this is what happens in post-hepatic jaundice. If you go by this, our case is not at all fitting into this. So our case is not post-hepatic. Our case is pre-hepatic. Okay, because our case had increased unconjugated. Urobilinogen was present, and AST, ALT, ALP were absolutely normal. So it's a case of pre-hepatic jaundice or hemolytic jaundice. Okay, fine. Let's see what happens in hepatic jaundice. See, in hepatic jaundice, so there will be normal cell turnover. So unconjugated bilirubin load is not very high in hepatic. Hepatic jaundice is liver is damaged. In hepatic jaundice, liver is damaged. So liver cannot conjugate. So conjugation will not happen. Okay, but because of the liver damage, the liver cells will swell. Okay, liver cells will get inflamed. Okay, when the liver cells get inflamed, the liver cells are surrounded by biliary canaliculi. So biliary canaliculi will get obstructed. So there will be intrahepatic cholestasis. You understand? So liver will try to conjugate, but because of the inflammation, the biliary canaliculi which is surrounding the liver cells will also get obstructed. So there will be intrahepatic cholestasis. So it will give you a mixed picture. So hepatic jaundice will give you a mixed picture. In these patients, both conjugated bilirubin will be high, unconjugated bilirubin will be high, both will be high. And urine urobilinogen will be decreased. It won't be very high, it won't be absent, but it will be decreased because conjugated bilirubin is not able to enter the intestine. And AST-ALT will be very high. Because of the liver damage, AST-ALT will be very high, and ALP will be mild to moderately increased. So this is a classical picture of hepatic jaundice. Okay. So if you see a kind of mixed picture, we see a kind of mixed hyperbilirubinemia. That is, both conjugated as well as unconjugated is increased. It's a case of hepatic jaundice. You understand? And the main clue to pick up hepatic jaundice is the enzymes. Enzymes will be over the roof. Okay. Enzymes will be very, very, very high. Okay. And the clue to pick up obstructive jaundice is conjugated will be high and ALP will be high. And the clue to pick up prehepatic is enzymes will be normal and unconjugated will be very, very high and urobilinogen will be positive. Okay, so this is how you should proceed with the diagnosis of jaundice. First thing you should see whether it's direct or indirect. First. Second, enzymes. Third, urobilinogen. Understand? So that's the thing. Okay, hope this is clear. You want me to repeat this or shall I go to the next slide? Okay, fine. Okay, so what is physiological jaundice? Physiological jaundice, we already know it's the most common cause of unconjugated hyperbilirubinemia. It usually happens in the babies because of their immaturity. Their liver is not that mature enough, so they cannot conjugate bilirubin. So whatever bilirubin that comes to their liver will remain unconjugated because of the it's usually seen in the preterm babies, sometimes the term babies also. It will resolve by itself. You don't want to worry about it. That's called physiological jaundice. Okay, rarely bilirubin will go beyond 5 to 6. Usually resolves within 2 to 4 weeks. Once the breastfeeding is started, okay, once you keep the baby for 2 days in phototherapy, it will be normal. So you don't want to worry about it. So that's the fourth option, physiological jaundice. Okay, fine. So last, this answer is prehepatic jaundice. Okay, 12th, which tissue or organ is least dependent on purine salvage pathway? Least dependent. What's the answer? This is slightly a tricky question. Least dependent, that is, this tissue doesn't need purine salvage pathway at all. Okay, I think most of you have got it wrong. Okay, let's see. See, liver is a major site for de novo synthesis of purines. Okay, liver can produce purine from the scratch. Liver can produce purine from the scratch. It has all the enzymes necessary for it. The rate limiting enzyme is called phosphoribosyl pyrophosphate glutamyl aminotransferase. It's PRPP glutamyl aminotransferase. It's called PGAT. Okay, this is a rate limiting enzyme of purine synthesis. Okay, this enzyme is present in the liver, but this enzyme is absent in few tissues. Okay, brain, leukocytes, red blood cells. So these tissues are completely dependent on salvage pathway because these tissues cannot synthesize purines de novo because they don't have the enzyme necessary for it. Okay, where liver has the enzyme necessary for it. Okay, so liver can synthesize purine for its own or for the rest of the body. Okay, however, brain, leukocytes, RBCs cannot synthesize because they don't have that one important enzyme called PGAT. So these three tissues are dependent on purine salvage pathway. Since liver has a capacity to produce a synthesis, produce a de novo purine synthesis, it is not dependent on purine salvage pathway. So the answer is liver. Liver is not dependent on salvage pathway because liver has all the capacity to produce synthesis, to synthesize purines from the scratch. That is called de novo. Okay, so liver is not at all dependent on purine salvage pathway. Okay, the three tissues which are dependent on purine salvage pathway are brain, RBCs, and leukocytes. They are dependent because they cannot synthesize purine de novo because we don't have that enzyme called PGAT. Okay, so what is a defect that can arise when that salvage pathway is defective? That's called Lesch-Nyhan syndrome. This is that syndrome. Okay, what's the classical picture of Lesch-Nyhan syndrome? Anybody? So how does Lesch-Nyhan syndrome will present? Yes, it has a classical picture of self-mutilation. Okay, so since salvage pathway is affected, brain cannot synthesize purine, so brain will not get the DNA. So brain cells will undergo damage. So these patients will have mental retardation. Their mental development will be delayed. They'll behave aggressively. They will have self-mutilation. They'll have hyperuricemia also. Fantastic. Okay, good. So the answer for this question is liver. Okay, so why liver? Because liver can produce or synthesize urine from the scratch, whereas other three tissues are dependent on urine salvage pathway. So liver is least dependent on urine salvage pathway. The answer is liver. Answer is D. Okay, fine. So what's the answer for this? High insulin glucagon ratio activates So whenever there is insulin levels are very high in the blood, what it activates? Correct. Good. Most of you got it correctly. Okay. Most of you got it correct. It's C. Let's see how. See, there are two states. Okay. Either you can be in any one of these state. Okay. You can be after fasting or you can be after a meal. That is called fed state. Okay. Fed state or fasting state. Okay. What is fed state? Fed state is immediately after the meal. Immediately after a meal, what happens to insulin levels? Will it be high or low? You take a meal, say breakfast or lunch or dinner. What happens to insulin levels in your body? Will it be high or low? Yes, it will be high. So that leads to a condition called high insulin glucagon ratio. Glucagon levels will be low. At the same time, when you are fasting, what happens? Your insulin levels will be low. Okay, fasting insulin levels should be low. Glucagon levels will be very high. So the insulin glucagon ratio will be low. Okay, so whenever there is high levels of insulin, gluconeogenesis is inhibited because insulin is a hypoglycemic hormone. It inhibits glycogenolysis also because both these two pathways, gluconeogenesis and glycogenolysis, that is production of glucose and breakdown of glycogen, will increase glucose levels in the blood. So both these will be inhibited by insulin. Okay, it will activate glycogenesis. Why? There are a lot of glucose in our body, so it will convert everything to glycogen and store it in the liver and skeletal muscle. So it activates glycogen synthase. That's the answer. Okay. At the same time, it inhibits lipolysis by acting an enzyme called hormone sensitive lipase. The exactly reverse will happen in the fasting state. Okay. Glucagon will promote gluconeogenesis. Glucagon will promote glycogen breakdown. Glucagon will inhibit glycogen synthesis. Glucagon will promote lipolysis. Okay. This is what exactly happens. Okay. So high insulin glucagon ratio, which you see after a fed state, will inhibit glucose production from glycogen and gluconeogenesis. So it inhibits those two pathways, but it will convert all glucose into glycogen by activating glycogen synthase, and it will inhibit lipolysis. That's why insulin is also called as anti-lipolytic hormone. Okay, so that's the thing. So answer will be glycogen synthase. High insulin glucagon ratio will activate glycogen synthase. Okay, that's the answer. Any doubts in this question? How does insulin activate or inhibit the enzymes? By phosphorylation or dephosphorylation? Even enzymes are covalently modified, they can be either active or inactive. One important covalent modification enzyme can undergo is called phosphorylation, right? Or dephosphorylation. Okay. What does insulin do? Will it phosphorylate or dephosphorylate? Correct. Good. It undergoes dephosphorylation. So it will dephosphorylate glycogen synthesis and makes it active. Okay, very good. Okay, so insulin decreases glucose by dephosphorylation. You remember like that. Insulin decreases dephosphorylation, something like that. Okay, fine. Okay, this is an interesting question. 14, 30-year-old men, unconscious state. What is this? What is this? Anybody? Even if you don't okay, there's no clue. Okay, you have to calculate it. Okay, fine, good. So it's metabolic acidosis, high anion gap. Okay, so with this case history, I'll also tell you how to approach a patient, how to approach or how to diagnose or how to crack the ABG. Okay, I'll tell you. So this is what I follow. Okay, you can follow if it is easy for you, or else you can follow whatever that is easy for you. Okay, see, step one. Okay, is there a problem in acid-base balance? That's the first question you should ask yourself. Okay, is there a change in pH? Yes, this patient's pH is 7.15. So what is it? Is it low or high? It is low, so it's acidosis. Okay, so this patient is having acidosis. The second important question is it metabolic or respiratory? How do you answer this? You look at the bicarbonate levels. Bicarbonate levels are reduced in this patient, so it is metabolic. Okay, if it is respiratory, what will happen to PCO2 levels? Anybody? So if it is respiratory acidosis, what will happen to PCO2? Fantastic. It will be very high. Okay. Is it high in this patient? No, it's not high. It's 20. It's not high. Okay. So it's predominantly metabolic. So it's metabolic acidosis. So whenever you see a patient with metabolic acidosis, the next step you should do is to calculate anion gap. Okay. So if you calculate anion gap in this patient, so anion gap is useful only in patients with metabolic acidosis. Okay, there is no point in calculating anion gap for patients with respiratory acidosis, respiratory alkalosis, or metabolic alkalosis. Okay, the only relevance of calculating anion gap is to find out the cause for metabolic acidosis. Okay, so here the anion gap is 32.5. It's very high. So it's a classical case of metabolic. So is it compensated? How do you say it's compensated or not? So bicarbonate is reduced. So what is the compensation you expect? See, compensation usually goes in the same direction. Okay. If you see a fall in bicarbonate, you should see a fall in PCO2. It's not vice versa. Compensation always goes in the direction of the primary defect. Okay. It never goes vice versa. So you need to see a fall in PCO2. Is the PCO2 low in this patient? Yes. How much it is? It is 20. Okay. So is it appropriately low? Is it compensated? How do you find out that? So there's an easy rule for this. It's called two-digit rule. Okay. So you see the last two digits of pH. What's the last two digits of this pH of pH of this patient? You just type in here in the chat box. What's the last two digits of pH of this patient? 15. So the answer, the expected PCO2 will be 15 plus or minus 5. So somewhere in between 10 to 20. So the patient's PCO2 is 20. So this is compensated. That's it. It's a very simple rule. That's what I use. Okay. This is called two-digit rule. There are a lot of other complicated formulas, but I don't think so we'll be able to remember those formulas in the exam. So I usually use this. It's called two-digit rule. Okay. So this patient's anion gap is high. It is compensated. Okay. So this is a metabolic acidosis with high anion gap compensated. Can you give me one cause for this? Which condition will cause metabolic acidosis with high anion gap? DKA. Very good. Okay. Diabetic ketoacidosis. Okay. Lactic acidosis. Very good. Okay. Very good. So good, 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 good. Okay. So there's a cause could be diabetic ketoacidosis. Okay. Fine. So what's the answer for this 15th MCQ? Calculate osmolality. What's the formula for calculating osmolality? It's a very simple formula. Okay. So 2 into sodium plus glucose by 18 plus urea by 6. Okay. So 2 into sodium is 140 into 2, that is 280. Glucose is 180. 180 divided by 10 is 10. Urea is 60. 60 by 6 is 10. So 280 plus 10 plus 10. So answer is 300. 300 milliosmoles per kilogram. Okay. So so that is 200. 2 into 140 plus 180 by 10 plus 60 by 6 is equal to 300. Okay. So that's the answer. The answer is 300. Okay. Fine. So this is how you calculate osmolality. It's a pretty straightforward, easy question. So answer is B actually. Okay. The answer is B. So what is osmolar gap? We saw about anion gap. What is osmolar gap? See, osmolar gap is nothing but the difference between the osmolality that you calculated and the difference between the osmolality you can measure by using some machines. Okay. So osmolar gap is nothing but measured osmolality minus calculated osmolality. Okay. So measured osmolality, there's an instrument called osmometer. You keep the blood sample into that meter, it will give you the osmolality. Okay. Or urine sample, it will give you the osmolality. Okay. So when you see the difference between these two, there should not be any difference. Okay. Usually they should be same. If you see some difference, apart from sodium, glucose, and urea, there is something unmeasured osmoles are there in the blood. You understand? Because we are accounting only for these three in the calculation: sodium, glucose, and urea, which are the most important osmotically active substances in the blood. Okay. If you see there is a gap, if you see there is a difference, that means there are some unmeasured osmoles in your blood. Okay, so this is used to identify some disorders, identify some disease conditions, especially the osmolar gap is increased in patients with alcoholism and ethylene glycol poisoning. Okay, so you cannot factor in alcohol here and you cannot factor in the poison here. So there will be some gap. Okay, so when you see an increase in osmolar gap, so there is something wrong in the patient, which could be due to an alcohol intoxication or it could be due to an ethylene glycol poisoning. Okay, so this is one important take-home message from osmolarity. Okay, you should know what is osmolar gap. You should know in which conditions osmolar gap will be increased. Okay, see, same concept. Anion gap is increased because there are some unmeasured anions. Okay, so like that, osmolar gap will be increased because of there are some measured. So there are some unmeasured osmotically active substances. Okay, so this you should remember. This can be asked in some MCQs. Okay, my answer is 300. Okay, fine. So very important, very easy question. This one, 16th. What is the answer? What's the answer for this question? Yeah, the answer is C. Okay, molybdenum is a cofactor for xanthine oxidase. Okay, so whenever this enzyme comes across, you should know one important fact. Okay, a drug which is targeted towards this enzyme is called allopurinol. Okay, so in which condition we use allopurinol? Anybody? Good. This allopurinol is used in the condition called gout. Okay, gout is characterized by hyperuricemia, that is increased serum uric acid levels, leading to deposition of monosodium uric crystals in the joints, leading to arthritis. Okay, so this allopurinol inhibits this enzyme called xanthine oxidase, and this inhibition is called suicidal. Why? Because see, this is a function of xanthine oxidase. It converts hypoxanthine to xanthine to uric acid. This is the normal function of xanthine oxidase. Okay, and for its action, it needs molybdenum as its cofactor. Okay, so we need molybdenum, only then xanthine oxidase can be functional. Okay, and this xanthine oxidase is inhibited by allopurinol. Okay, how? Why it's called suicidal? Because allopurinol will be acted upon by this enzyme. Okay, the enzyme commits a suicide here. Okay, enzyme instead of acting on hypoxanthine, it acts on allopurinol because both look structurally similar. Okay, so it converts this allopurinol to form alloxanthine. Okay, and this alloxanthine can go and inhibit xanthine oxidase by binding to its active site. Okay, so enzyme commits a suicide here. That's why it's called as suicidal inhibition. Okay, fine. So that's about it. Okay, so the answer is xanthine oxidase, and it needs molybdenum as a cofactor. Okay, fine. So let's go to the previous slide. I will ask you one question. So what's the cofactor for alcohol dehydrogenase? Which mineral acts as a cofactor for alcohol dehydrogenase? A B1 is correct. B1 is not that, it's not directly connected to it, but yes, zinc. Zinc is a cofactor for alcohol dehydrogenase. Carbonic anhydrase. Zinc. What is the important function of carbonic anhydrase? What is the important function of carbonic anhydrase? Just type in the answer. Yes, it is an important enzyme involved in bicarbonate buffer system. Okay. Superoxide dismutase. What's the mineral that is a small that will act as a cofactor? Superoxide dismutase. It is an enzyme which is involved as an antioxidant. Okay. The answer is superoxide dismutase requires three minerals: copper, zinc, manganese. Okay, copper and zinc because it has cytochromes. It also has manganese. Okay, so these are the various. These are the options. These are the answers for the other options in the question. Alcohol dehydrogenase requires zinc. Carbonic anhydrase requires zinc. Okay, Superoxide dismutase, which is an antioxidant enzyme, requires copper, zinc, manganese, and xanthine oxidase requires molybdenum. Okay, fine. So answer is xanthine oxidase. 17. So this is an interesting question. What's the answer? Good. Thiamine. So what's this condition actually? Anybody can name this condition? What is this condition? The answer is good. What is this condition? Good. This is Wernicke's and Korsakoff syndrome. Okay. So what happens? So patient comes in, he's an alcoholic. Okay. So he's confused. He has ataxia, he's not able to walk properly. His eye movements are restricted. It's called ophthalmoplegia. So the intern checks the blood glucose, found to be hypoglycemic, so immediately gives dextrose. But the condition is not improved. It is getting worsening. Why? We'll see. Okay. He thinks this could be hypoglycemic, or she thinks it's hypoglycemic and gives dextrose. That's what people do, right? When you see somebody with low glucose, you infuse glucose. Okay, and you expect them to get better. But in this patient, he is getting worsened. Instead of getting better, he's getting worsened. The condition is getting worsened. Why? So exactly, okay. Whenever an alcoholic presents with these symptoms, so like ataxia, ophthalmoplegia, and hypoglycemia, is more likely to this syndrome called Wernicke-Korsakoff syndrome. Okay. See, if you give only glucose, he's going to deteriorate. He's not going to improve. Okay. You have to give glucose along with thiamine, because just giving glucose alone is not going to help, because of lack of thiamine, you give only glucose. Thiamine is a cofactor for various enzymes, pyruvate dehydrogenase, alpha ketoglutarate dehydrogenase. These are the two important enzymes of carbohydrate metabolism, especially glucose. Since these two enzymes are inactive, your glucose cannot get metabolized. Okay. Because of this, there will be accumulation of lactose. All glucose will be converted to lactate, okay, and glucose. And this is going to be bad. Brain cells will get damaged because of lactic acidosis. So whenever you see a patient with alcohol intake with all those neurological features, along with hypoglycemia, don't just give glucose. Okay. You have to give glucose along with thiamine, because alcoholic patients are more prone to thiamine deficiency because they won't have proper food. One, now, alcohol inhibits thiamine absorption in the intestine, so they'll be thiamine deficient. So just by giving glucose alone, they will not be able to metabolize that glucose at all. Instead, the glucose will be diverted to form lactate, which causes acidosis, which is damaging to the brain cells. So the condition will worsen. Instead of expecting an improvement, you will see a worsening there. So it's always advised to co-administer glucose along with thiamine. Okay, only then you can correct or reverse this Wernicke-Korsakoff symptoms to a minor extent. Okay, if the brain damage sets in, we cannot reverse it. Okay, but to a minor extent, you can reverse it by co-administering glucose along with thiamine. So the answer is thiamine. Okay, so this condition is called Wernicke-Korsakoff syndrome. Okay, fine. So what is the answer for this true statement regarding dietary fiber? What's the answer for this? Correct. Okay, it is dietary fibers are not digestible completely because we don't have enzymes to digest them, because they're made up of cellulose. Okay, so we cannot digest them. They are partially digestible. Okay, because we don't have enzymes. And these are the various dietary fibers containing foods, oats, vegetables, fruits, legumes. So if you see here, whatever cellulose that goes inside comes out of cellulose. So we cannot digest cellulose because we don't have enzymes. Okay. So what is its function? If it is not going to be digested, it is not going to give any energy. So why do we take it? Okay. One thing, it gives you early satiety. It gives you fullness early. So you don't have any caloric value. Okay. So it gives you fullness, so you will stop eating more. If you take dietary fibers, you will not eat more, so it's a good thing, right? So we will not eat over calories. Okay, sure, 8:30 I'll finish, don't worry. Okay, fine. So it provides feeling of early satiety. Okay, so there's no caloric value. It adds roughness to the stool, so there won't be any constipation. That's one important thing about dietary fiber, since it prevents constipation. Okay, it binds bile acids and cholesterol, so bile acids cannot be absorbed. So bile acids will come out in feces, so all cholesterol will be converted to bile acids. So cholesterol levels will come down. Second, it also improves glucose tolerance. Okay, so these are the various functions of dietary fibers. It provides early satiety, prevents constipation. Okay, it binds cholesterol and bile acids and prevent their absorption. So cholesterol will come down. It also improves glucose tolerance. Okay, fine. So the answer is it increases roughage to the stool. Okay, what is the answer for this? Which of the following drug acts on the peptidyl transferase and inhibits it? Correct. It's chloramphenicol. Okay, fine. So you already know that. So I had a video, but I think you already know translation. So I will skip this video. So these are the various inhibitors of translation in prokaryotes. Okay. See, translation has in general has four steps. Okay. Translation initiation, transfer of peptide bond, then translocation. Okay. So streptomycin inhibits the formation of initiation complex itself in the prokaryotes and telling, that is in the bacteria. Okay. So it's bind to 30S subunit and prevents the formation of initiation complex. Okay. Tetracycline prevents the binding of tRNA to the A site. That's a second step. You have to find the complex, form the complex. Then the amino acid tRNA should go and bind to the A site. Okay, that is inhibited by tetracycline. Chloramphenicol will inhibit the transfer of peptide bond. That is peptide transferase activity in the prokaryotes. Okay. Then erythromycin will inhibit the translocation. That's the last step. Okay, it has to move from A to P. Okay, that will be inhibited by erythromycin. Okay, these are the inhibitors of translation in prokaryotes. Okay, what are inhibitors of translation in eukaryotes? Same thing. Puromycin inhibits the binding of amino acid tRNA to the A site. So this is eukaryote. That is prokaryote. This is eukaryote. Okay. So binding of tRNA to the A site is inhibited by puromycin. Cyclohexamide is a counterpart of chloramphenicol. It inhibits peptide transferase in the eukaryotes. Okay. So CC, chloramphenicol in prokaryotes. Cyclohexamide does the same thing but in the eukaryotes. Diphtheria toxin also inhibits translocation. Erythromycin in prokaryotes, diphtheria in the eukaryotes. So that's about the thing. So the answer is chloramphenicol, 19. Chloramphenicol. Okay. 20. I think you already know this. Shortening of the very end of the chromosome during each replication is prevented by telomerase. Very good. Okay. This telomerase is actually a very important enzyme. Okay. See, this is what happens in the DNA replication. So one strand will be continuously synthesized, that is towards the replication fork. Okay. The one strand will be discontinuously synthesized, that is called lagging strand. This is leading strand, this is lagging strand. Okay. You see this leading strand, this green color, what you see is RNA primer, that will be removed. So this 3' end is not adequately replicated. Okay. So this is the ends of the chromosome. Okay, so this 3' end, whenever a cell divides, this 3' end gets shortened. Every time when a cell divides, it gets shortened. So shortening, the DNA cannot replicate further, so cells cannot divide further. Cells will reach a stage called senescence. Okay, the cells will die eventually. So how do we prevent this? So we have to maintain the length of the DNA at the ends. That's the telomeric ends. So this is a centromeric end, and this is a telomeric end. So we have to maintain the length of the DNA at the telomeric end. Okay, that is done by this enzyme called telomerase. Okay, this telomerase has three important components. Okay, it has a protein and it has an RNA in itself. Okay, it's called telomeric RNA. Okay, that protein has a polymerase activity. Okay, so thus polymerase is nothing but RNA dependent DNA polymerase. Do you know any other RNA dependent DNA polymerase? Have you come across any other RNA dependent DNA polymerase? It's found in HIV. What is it? RNA dependent DNA polymerase? Correct. That is called reverse transcriptase. Same thing. This telomerase also has an RNA-dependent DNA polymerase. So this telomerase complex itself will have an RNA. Okay. Using this RNA, telomerase will synthesize the DNA so that the length of the DNA at the telomere can be maintained. Okay. So that's how telomerase helps in maintaining the length of the DNA at the ends of the chromosomes. Okay. That is called telomeric DNA. The enzyme is called telomerase. Okay. And this enzyme is very, very important because this enzyme is targeted by certain chemotherapeutic drugs because cancer cells will have this enzyme in huge amounts. The cancer cells will proliferate in an uncontrolled manner. Okay. So telomerase expression is very high in patients with cancer. Okay. So telomerase is actually a good target by many chemotherapeutic drugs. Okay. So there are many chemotherapy drugs recently it's in clinical practice where telomerase has been targeted because telomerase activity is very high in cancer, so the cancer cells can divide rapidly. Let's go to the next slide. Okay, what is the answer for this? An amino acid can have more than one codon. What is the answer for this? Which property of genetic code can explain this? Correct. This property is called degeneracy. Okay. What is degeneracy? See, there are only 20 amino acids in our body. Okay. But how much codons we have? We have 61 codons. Okay. So that itself says an amino acid can have more than one codon. Okay. So this property is called degeneracy, and this is possible because of something called Wobble's hypothesis. If you see the figure here, the first two base pairing will follow traditional base pairing. G can bind with C, A can bind with U. This is called traditional pairing. Okay. But in Wobbles, a third nucleotide will not follow traditional base pairing. It can bind with anything. Okay. So only the first two amino acids, first two nucleotides matter. Okay. This is called wobbling. Okay. And this wobbling will enable an amino acid to have multiple codons. Okay. So that's called degeneracy. What's the reverse of it? If I give you a codon, it can code only for one amino acid. So what property is that? If I give you an amino acid, you can give me multiple codons. Okay, I'm not asking you that. If I give you one codon, it can code only for one amino acid. So what property is that? Yes, correct. That is unambiguous. Okay. So given a specific codon, it can code only for specific amino acid. Okay. So that's about it. Okay. So what is universe? What is non-overlapping? See, codons are always read continuously. Okay, there is no comma, there's nothing, there's non-overlapping. And it is called universal, but not always, because there are few differences with mitochondrial genetic code. Okay, UGA, which codes for stop codon in the nuclear genome, will code for tryptophan in mitochondria. That's why they say it's not always universal. Okay, that's why they say that now recently they say that you should not call them universal, you should call them as standard. Okay, because there are some differences mitochondrial genome. Okay, UGA is actually a stop codon in the nuclear code, but the same UGA will code for tryptophan in mitochondria. So it's not universal. Okay. So AG and AGG, which codes for some amino acid in the nuclear, that is arginine, but it is a stop codon in the mitochondria. So there are some differences between mitochondrial genetic code and the nuclear genetic code. So it is always not good to use a term called universal. Instead, they are suggesting that you should use something called standard. Okay. So it's not universal because there are some differences between mitochondrial genetic code and the nuclear genetic code. Okay, let's go to the next question. So amino acid which contributes to the formation of serotonin. Correct, tryptophan. Okay, so it's a very simple question. Okay, so tryptophan can form three important compounds: serotonin, niacin, melatonin. Okay, three important compounds. Arginine can form three important compounds: creatine, nitric oxide, polyamines. Tyrosine, thyroid hormones, catecholamines, melanin. This is a very simple question, so I'm not going to discuss further. Threonine can form glycine. Okay, threonine can form another amino acid called glycine. Okay, that's it. Fine, so it's a very simple question. 23. Okay, this is an interesting question. A disorder causing excess of this amino acid leads to morphonoid features. What disorder is this? Correct. Homocystinemia. Okay, so this is the equation, which this equation will give you answers for multiple MCQs. Okay, homocysteine has to be converted to methionine by an enzyme called methionine synthase. For this, you need two important vitamins, okay, B12 and folic acid, okay, methylcobalamin and methyl tetrahydrofolate, okay. And homocysteine can also be converted to cystathionine by an enzyme called cystathionine beta synthase. For this, you need one more vitamin called B6, okay. So three vitamins are important for metabolizing homocysteine. Two for converting homocysteine to methionine, one for converting homocysteine to cystathionine, okay. So if any one of these enzymes or if any one of these vitamins is defective, is deficient, or the enzyme is defective, it leads to a condition called homocystinemia. Okay, there's one more important point here. What happens if B12 is deficient? What happens to folic acid levels? What is that condition called? If B12 is deficient, what happens to this folate? What is that condition called? Correct. It leads to something called folate trap. Okay, because all folate will exist as methyl tetrahydrofolate. Okay, so all folate will be trapped as methyl tetrahydrofolate. Okay, it's called methyl tetrahydrofolate trap. Okay, so if any of these vitamins are deficient, homocysteine will be elevated. Okay, and this homocysteine can lead to this morphonoid features. That is, the patients will be very tall, very thin. They have hyperextended fingers. Their joints will be hyperextensible. Okay, and they'll have scoliosis. Okay, and their lens will be dislocated. It's called ectopia lentis. And these features are called as morphonoid features. So homocystinuria can lead to morphonoid features. Okay. One more important thing, homocystinuria or homocystinemia is a cause for increased risk, is a cause for heart attack, especially in young people, because it increases the risk of thromboembolism. Okay. That's why in recent over last 3 to 4 years, there is a rapid increase in testing for homocysteine levels in blood. Okay, so hyperhomocystinemia can lead to heart attack, especially in young people. Okay, that's one more important point about this. Okay, fine. Last question: which hormone is usually estimated in the early morning for diagnosis of the disorder associated with adrenal gland? So, what disorder we are talking about here? So, adrenal gland disorders can adrenal gland. See, metanephrin is not a correct answer. Okay. See, adrenal gland has two things: cortex and medulla. Okay. Cortex dysfunction can lead to hypercortisolism, which is called as Cushing syndrome, or Addison's disease, which is adrenocortical insufficiency. Medulla can, there is one tumor which can sit in the medulla, which can lead to increased secretion of catecholamine. Sorry, there's a tumor which can sit in the adrenal cortex, which can increase the release of catecholamine. It's called pheochromocytoma. Okay, these are the three things we are going to deal with: Addison's disease, Cushing syndrome, pheochromocytoma. Okay, in order to diagnose Cushing syndrome, we need two things. What are the two things apart from cortisol? Anybody? If you want to diagnose somebody with Cushing syndrome, we need to test for two things. What are the two things? Cortisol and ACTH. Very good. Okay, cortisol and ACTH. Pheochromocytoma is catecholamines, metanephrines, and BMA. I am not going to talk about that. If you want to diagnose Cushing syndrome, you need three things: cortisol and ACTH. Okay, and this cortisol and ACTH will exhibit diurnal variation. As you could see from this figure, there is a good peak in ACTH and cortisol in the early morning, especially in the 8 o'clock range. Okay, so you have to get blood sample from this time. Only then you can get a good peak, good concentration in the blood circulation. Okay, if you estimate in other points, they are already low, you'll get falsely high or falsely low values. So it's advisable to take a blood sample for ACTH or cortisol only in the early morning. Okay, it's called early morning cortisol or early morning ACTH. Okay, one good sample. Okay, so that is the thing. See, metanephrines does not exhibit diurnal variation. Metanephrines comes from catecholamines. Catecholamines are secreted in burst, pulsatile fashion. Okay, so they won't exhibit diurnal variation. So if you want to check for something which is secreted in burst, you should collect 24-hour urine sample. Okay, for metanephrines, early morning sample will not help. Okay, so if you want to diagnose pheochromocytoma, you have to get urine sample collected over 24 hours. Okay, because it's secreted in the pulsatile fashion, not the burst. This frequent burst will be there. Okay, so metanephrine should not be checked in one sample. Okay, you have to collect a 24-hour urine sample and you check for metanephrin and catecholamines in that urine sample. Okay, so the correct answer for this question is cortisol. Okay, so cortisol is usually taken from the blood sample, which is collected early morning because cortisol and ACTH exhibits diurnal variation. Okay, so this is mainly done to rule out Cushing syndrome. Okay, and that's the answer for that question. Okay, so the last MCQ, the enzymes mostly involved in phase 1 of xenobiotic metabolism. What is the answer? Exactly. The answer is cytochrome P450 monooxygenase. So I'll put it in a very simple way. If you go to the next slide, you'll understand. See, xenobiotic metabolism happens like this. Okay. First phase converts a xenobiotic into a reactive metabolite. Okay. It makes the xenobiotic more reactive by putting a hydroxyl group into it. Okay. The second phase will conjugate the reactive metabolite and make it water soluble. So everything will come out in urine. Okay. These are the two important phases of xenobiotic metabolism. Phase 1, it adds a hydroxyl group and makes it reactive. Phase 2, it adds a conjugation, it adds a conjugate and makes it water soluble so that everything will come out in urine. Okay, these are the two phases of xenobiotic metabolism, phase 1 and phase 2. So phase 1 is nothing but adding hydroxyl group. Okay, this is mainly done by one group of enzyme called cytochrome P450 monooxygenase. Nearly 50% of the drugs is metabolized by this enzyme, cytochrome P450 monooxygenase. Okay, and this cytochrome 450, few points about this. It's a heme containing enzyme. It's present mainly in the liver. It is present in the smooth endoplasmic reticulum. It is also involved in the synthesis of steroids, bile acids, prostacyclins, thromboxanes, and vitamin D. Okay, so it's a very important group of an enzyme. So this enzyme plays a very important role in the phase 1. Okay, what is phase 2? Phase 2 is conjugation. We already know glucuronic acid addition. It's called glucuronidation. Sometimes you can add sulfate and make it water soluble. You can add glutathione, you can add acetyl group by acetyl transferases. You can add methyl group by methyl transferases. So by all these things, you can conjugate a metabolite and make them water soluble. If you make them water soluble, they will come out in urine. Okay, so that's how xenobiotics are metabolized and pushed out in urine. Okay, so phase 1 and phase 2. Phase 1 is hydroxylation, thereby it makes it more reactive. Phase 2 is conjugation, which makes them more water soluble so that they can come out in urine. So what are things that can go and conjugate? Glucuronic acid, sulfate, glutathione, acetyl group, methyl group. Okay, so the name goes like that: glucuronyl transferase, sulfatyl transferase, glutathione transferase, acetyl transferase, methyl transferase. As the name goes by. Okay, so that's the thing. So the answer for this question will be cytochrome P450 monooxygenase. That's the thing. Okay, I think I have completed all 25 questions. Okay, so any doubts? Any doubts? Hope it was useful for you. Okay, thank you. 1 minute. Okay, thank you for patient listening. Okay, hope we'll see soon. Thank you very much. Thank you. Okay, I'll end this meeting. Thank you.